speaker in this section, and I'm going to be talking to you about my work on X chromosome regulation in the Drosophila germline. So, as you all probably know, the classical model of sex chromosome evolution says that sex chromosomes evolved from ancestral autosomes that acquired a sex-determining locus, here resulting in a female with 2 X chromosomes and a male with an X and Y chromosome. Over time, sexually antagonistic alleles can arise, which provide selection pressure for inversions to occur, which suppresses recombination between the X and the Y chromosome. Over time, this leads to the complete loss of recombination between the X and the Y chromosome, and the loss of recombination along the Y leads to its degeneration, which results in the evolution of heteromorphic sex chromosomes. One consequence for the evolution of heteromorphic sex chromosomes is the evolution of sex chromosome-specific forms of regulation, such as dosage compensation or meiotic sex chromosome inactivation. I'm not going to be talking today about either of these, but of another form of X chromosome-specific regulation that we're calling X suppression. So X suppression is the observation that when a transgene is inserted onto an autosome, you see very strong, robust expression in the testes, but when the exact same transgene is inserted onto the X chromosome, you see a significant reduction in expression. And these transgenes have been assayed from multiple inserts across the melanogaster genome, showing that the entire X chromosome is transcriptionally suppressed, and it's this transcriptional suppression that we are calling X suppression. And while these transgene data do suggest that the entire X is transcriptionally suppressed, these data are limited. So to date, all evidence for X suppression comes from transgenes, which are modified transposable elements. So it could be that X suppression is a way to regulate X-linked transposable elements. Additionally, every single transgene that shows evidence for X suppression has used a testis-specific promoter, so it's also unclear if X suppression is specific to testis-specific genes. So the first thing I'm going to do today is characterize X suppression and specifically ask if it is transposon specific and if it is testis-specific. But for anybody in here who's worried that I'm just going to give you a purely mechanistic talk, I promise there is going to be some evolution, and I'm going to show you evidence that it looks like the X chromosome is actually adapting in response to X suppression. So just hang on with me while I characterize X suppression. So I designed an experiment to answer both of these questions. So what I did was I moved a small piece of the X chromosome to the autosome. These are small regions that contain very few genes, and what this allows me to do is directly compare gene expression for the same gene when it's located on the X chromosome and when it's located on the autosome. So I just want to point out that this experiment did control for dose, so there's only one copy on the X and the autosome. And I also want to stress that this was done without using transposable elements. So these are endogenous genes that we're assaying, so we can directly ask whether or not X suppression is transposon specific. Also, every region that has been moved contains both testis-specific and non-specific genes, so that I can look at how these two classes compare to directly ask if X suppression is also testis-specific. So, and I use qPCR to look at gene expression. So when we look at the two testis-specific genes in this transposition with expression on the Y chromosome, we see that both show a significant increase in expression when the gene is moved from the X chromosome to the autosome. And these data are exactly what was seen for transposable elements, which suggests that X suppression is not transposon specific. And when we look at the non-specific genes, we see that they show the same pattern, that both genes show a significant increase in expression when they move from the X chromosome to the autosome, which suggests that X suppression is not testis-specific. I've now assayed multiple independent transpositions that have moved different regions of the X chromosome to the autosome, and within each I've assayed multiple genes. And when we compare autosomal expression relative to X-linked expression, what we see is that non-specific genes show an approximate 2-fold increase in expression when they move to the autosomes, and testis-specific genes show an approximate 4-fold increase in expression when they move to the autosomes. And so while we can say that X suppression is not transposon specific and it's not testis specific, we do see a significant difference in the magnitude of effect size between the non-specific and testes-specific genes, which suggests to us that the promoters of testes-specific genes are particularly strong, so that when a testes-specific gene moves to an autosome, you see an even greater increase in expression. So we have an additional line of evidence that suggests that X-linked testes promoters are particularly strong, and this data comes from new genes. So these are new genes that could have arisen by retrotransposition or DNA-based duplication, and we bin these genes based on their age when they arose on the phylogenetic tree, and then we looked at the expression of these genes in melanogaster testes. And what we found is that for a new gene that finds itself on an autosome, you see pretty high expression, and over time that doesn't really change, but we see a different pattern for new X-linked genes. A new naive X-linked gene that finds itself on the X chromosome is in a really suppressive environment, and you can see that its expression is suppressed, but over time X-linked genes increase their expression in the testes, suggesting that these genes are evolving in response to X suppression to increase their expression, and again, it's suggesting that these promoters are evolving to become even stronger. So next we asked whether we could identify a regulatory element in the promoters of testes-specific genes on the X that could be mediating this expression, and we took the promoter region that immediately flanks the transcription start site, and we computationally identified a candidate regulatory motif. When we look in highly expressed testes-specific genes, we find that this motif is found over 50% in over 50% of all X-linked genes. And when we look for this same motif on autosomes, we find that it's found at 18%. So it's found roughly 3 times more frequently in X-linked genes than autosomal genes. And when we look for the same motif element in housekeeping genes, so these are genes that are broadly expressed throughout all tissues, we find that it's rarely found in 5 to 7% frequency. So these numbers were really exciting and suggestive to us, but of course this was a computationally identified motif element, so we wanted to find additional lines of evidence to convince us that it was real. So the first thing that we did was we looked at the effect of this element on expression. So when we look at the effect of expression for autosomal and X-linked testes-specific genes, promoters that have this element, we see a significant increase in expression for both of these classes, suggesting that this motif is mediating increased testes expression. We also looked at where this motif is located relative to the transcription start site, and what we found is that it's located immediately upstream of the transcription start site, and when we looked in housekeeping genes, we found no such pattern. Okay, so no doubt you've noticed when I showed you this motif element that all the letters are different sizes, and the letter height is a rough approximation for the frequency of a particular nucleotide at a position. So really low letter height suggests that any of the 4 nucleotides are found at equal frequency at that location, but a really tall letter height like this C suggests that every time we see that motif, we always see a C there, which suggests that that would be under strong constraint, which is what we looked at. So we looked at divergence of this motif between melanogaster and Incuba relative to letter height, and we find a highly significant negative correlation between divergence and letter height, suggesting that this motif is under evolutionary constraint between melanogaster and Incuba, which suggests to us that the more important letters are under functional constraint. And when we looked in housekeeping genes, again, we did not find this pattern. So, taken together, all of these lines of evidence suggest that this motif is real and is mediating an increase in testes-specific expression, but we really wanted to validate that, and we did that using transgenes. So we used the promoter CG12681, which I already showed you, shows evidence for X suppression, and whose promoter contains one of these candidate motifs. And we used the promoter region to drive LAC-C in the Drosophila testes. And for our wild-type controls, we do see a significant downregulation of expression on the X compared to the autosome, which is exactly what we expect from our controls, because I already showed you that this gene is under X suppression. But what we really wanted to know was what happens when we break this regulatory element and how it might relate to testes expression. So we mutated these two C's to A's to see what happens. And what we find is that we find a significant difference in expression, a significant decrease in expression for both the autosomal transgene and the X-linked transgene. So what these data show is that we did find a regulatory motif that is significantly over-enriched in X-linked testes-specific genes that does mediate an increase in testes expression. So, just in summary, what I showed that the X chromosome is suppressed in Drosophila testes, and using multiple transpositions, I was able to show you that testes-specific genes show an approximately 4-fold increase in expression, while non-specific genes show an approximately 2-fold increase in expression when moved to the autosomes. I showed that the X chromosome appears to be adapting to X suppression, that new X-linked genes evolve and increase expression in the testes. We identified a regulatory motif that is enriched in X-linked testes-specific genes. It's associated with higher expression in the testes. It's located directly upstream of the transcription start site. It's functionally constrained, and we validated that in